Okay, everyone, let's get started talking about catabolism today. So, in lecture on Thursday, I introduced you guys to this idea of metabolism, right? We talked about how metabolism is the sum total of all the chemical reactions taking place inside of a cell. And then I spent a fair bit of time with you talking about how enzymes pull off the tricks that they do as catalysts. The bottom line is they reduce a reaction's activation energy. Right? So it makes it easier for substrates to get to an excited state, to achieve that transition state that they need to pass through on their way to becoming products. They don't change the Gibbs free energy for the reaction. They can't change the Gibbs free energy of a reaction. They can only lower the activation energy. And they can only help reactions take place that have a favorable change in Gibbs energy, meaning you go from high energy to lower energy through the course of the reaction. Right? So that's how enzymes do their thing. And again, metabolism is a lot of stuff, right? There are hundreds, maybe a couple thousand chemical reactions that a cell will catalyze or help along at any given point in time. That's what makes us different from non-living things, is we have those reactions taking place within us, whereas in the absence of those catalysts, they might not. Right? So it's a pretty large topic. It's not a completely incomprehensible area of study, right? A couple hundred, maybe a few thousand chemical reactions. It's the sort of thing, if you really put your mind to it, you could start after a year or two, perhaps, a study to say, I got metabolism down. I know the whole thing. So it's comprehensible, but it's not something we have to worry about too much for a class like this. We just want to keep an eye on the big picture. That's the scale of things. Those are the general rules that are involved in the game, and how it is that living things work within those rules to game the system a little bit and let us do things that are sort of special compared to what would happen if we didn't have enzymes, biological catalysts, to help us out. Today, what I want to do is focus on one part of those reactions of metabolism, and we're going to focus just on the part that we call glycolysis. Particularly, that's how we extract energy from glucose, is we're converting it to another sugar called pyruvate. We start with a 6-carbon sugar. Picture that, 6 carbons linked together. We start with a 6-carbon sugar, and at the end of glycolysis, we have 2 3-carbon sugars. Okay? That means some energy is going to be released, because at the very least we're breaking one carbon-to-carbon bond, right? And you guys know that those are energy-rich bonds. When we release that energy, there should be an opportunity to capture some of that energy. And in fact, what will happen is you'll see that we get a net gain of 2 ATP molecules for every glucose molecule that we convert to pyruvate molecules. Okay, so that's the big picture of what's going to be happening today. In a word, we can divide metabolism up into 2 categories. One of those categories is catabolism, catabolic processes, and the other category are anabolic processes, or anabolism. All right, catabolic processes are the ones that break things down. Okay, you go from big molecules to smaller molecules. Anabolic processes are the ones that build things up. So you've probably encountered the word anabolism or anabolic before. The only time I seem to hear it in the newspapers and things like that is in the context of anabolic steroids. Those are the kind of steroids that people take to get bulked up and perform better in athletic competitions. Anabolic steroids are the steroids that literally bulk you up, right? They build you up from small and puny to big and huge and strong and such. Nobody takes catabolic steroids, the ones that make you weaker and less muscular, but for every anabolic steroid, there is actually a catabolic steroid, and there's sort of a balance between the two of them within our bodies. But I'm getting off topic. the thing today is we're going to talk about like the cornerstone of catabolism, of catabolic processes within cells. Today we're going to talk about glycolysis, breaking glucose down into 2 3-carbon sugars called pyruvate. So that's what's up for today. And I'll tell you right at the onset, I see in the lecture notes, this is front and center in the beginning of today's lecture notes. And actually, I also noticed that today's lecture notes are like 4 pages long for not just today's lecture, but also Thursday's lecture. Here's the thing. This stuff is easy for me to talk about, right? There's not a lot of challenge for me to talk you through these things. But I'll tell you right up front, what I'm going to talk with you about today might be high. It's high on the list of the things that might take some extra work on your part. I'm going to show you the ropes today, but to really get comfortable with what's going on, I think you need to spend some extra time wrestling with these things. I'm going to tell you, I'm going to point out to you along the way. Here's an important word, here's an important word, here's an important word. Check out this scheme of things that's going on here. But I think after the lecture's over, this is one of the kind of lectures where I think you need to spend some time just, looking at the pictures, maybe reading the textbook, wrapping your head around it and making sure you're comfortable with these things. I'll tell you the highlights along the way and point to where the important stuff is going on, but to really wrap your head around it, I think you need to spend some extra time with it. So, some truth in advertising at the beginning here today. This is easy for me. I think that might mean it's going to be a little bit harder for you just because you guys have to do some of the lifting for this lecture. But again, today we're talking about a subset of metabolism. We're specifically talking about the cornerstone of catabolism. And even more specifically, I'm going to focus on glycolysis, breaking down glucose to pyruvate. That's actually a 10-step process. So there are 10 enzymes that are involved. And during the course of today's lecture, I'll introduce you to each of those 10 enzymes. All of the chemical reactions that I'm going to be talking with you about today, all of the chemical reactions we'll be talking about today are these redox reactions. You might remember from Thursday last week, I told you that a redox reaction is one where you're moving electrons around. Okay? That's the 30,000-foot summary of what a redox reaction is. Let's get down to 20,000 feet to talk about redox reactions. And then I'll get into talking with you about, ultimately, glycolysis. So, and before I do that, let me give you this just to tie in what I'll be talking with you about here today with what you got to see in the video before class today. All right? I asked a couple people. For those of you who were here before class started, we played a YouTube video of some guy blowing stuff up with hydrogen. Right? It's kind of cool. All right? That guy, to me, when I first saw that video, I was thinking, what is the Speaker of the House doing blowing stuff up? Did you guys notice a similarity between the guy who was in the video and Paul Ryan, the current Speaker of the House? I was like, no, that can't be him. But I don't know. Maybe he's moonlighting, doing something else on the side. But I wanted you to see that video. At least it's moderately entertaining to see the guy's excitement at blowing things up. I had never encountered anybody who opened a Pringles can quite the way that he does. That was a neat thing to see. I've got a Pringles can at home. I'm thinking I got to find some hydrogen now to see if I can't replicate that myself. It looked pretty fun. But the whole point of that guy's video, and the reason I thought it might be fun for you to see it, aside from the entertainment value, is it illustrates very graphically what I've got in this tired old slide that's on the screen right now. And that is that, when we're talking about these kinds of reactions, particularly here, the reaction on the left side of the screen, going from hydrogen gas and combining that with oxygen gas, you're releasing a lot of energy, right? Check out those molecules at the top left part of the screen, right? The H2, that's hydrogen gas. You've got a covalent bond, just one covalent bond between two atoms with the same affinity for electrons, right? That is like the definition of a high energy bond, right? No quibbling about it. When you guys are thinking about high energy bonds, that's got to be a bell ringer. It's not going to be, you can't do better than that. Two atoms with the same affinity for electrons, sharing electrons, that's a high energy bond, period. In oxygen gas, it's the same story. We're talking here about oxygen atoms with the same affinity for electrons, sharing electrons, now two covalent bonds. Those are high energy bonds. When you break those bonds, either the hydrogen to hydrogen or the oxygen to oxygen bonds, when you break them, you should expect to release energy. High energy bonds, when they get broken, release energy. Can't be much simpler than that. And when you release that energy, you can release it as a big explosion, like the guy in the YouTube video was experiencing as he was opening his can of Pringles. As you go from substrates, hydrogen gas and oxygen gas, to product, water, right? You could release it as a big explosion. Or you could do it in a stepwise fashion, where you have the same end result. You go from hydrogen gas and oxygen gas to water, but along the way, instead of having a big flash, a big explosion of release, explosive release of the energy, you do it a little bit at a time. You do it in a stepwise fashion and on a controlled release so that you can capture some of that energy. The end result's the same. You release the same amount of energy. The delta G is the same. The difference is uncontrolled explosion. Probably not a good thing to have happening inside of our stomachs or inside of our cells, right, for obvious reasons, or a controlled process whereby we can capture some of that energy and use it to drive other chemical reactions. Guess which one our body's evolution has brought us to? We don't have very much of the spontaneous combustion kind of stuff going on. We've got all the time, every second, pretty much every cell is doing what's on the right-hand side of the screen. This stepwise release of the energy, going from high energy to lower energy, negative delta G, but in a controlled fashion. That control is what it is we'll focus on today when I talk with you about glycolysis. All right, it's a stepwise release as opposed to a big poof and all of the energy being released at one time. Now, somewhat interestingly, this is an example of a redox reaction. Let's take a closer look at a couple redox reactions. Again, I mentioned on Tuesday and just a few minutes ago now that a redox reaction, by definition, is one where electrons are being moved from one thing to another. Get a little bit more detailed, focus on it a little bit more. Let me point out to you that sometimes it's really easy to see that something's a redox reaction because you can literally, at least on paper, see the electrons getting moved from one place to another. Consider sodium and chloride. Right? Sodium chloride, that's salt. It's an ionic bond. We've talked about this a couple of times already in this class. Sodium has that one extra electron in its outermost orbital. It's really happy to dish that off onto something else to be involved in a redox reaction, to get rid of its electron, to move that electron somewhere else. And chloride has that circumstance where it's just one electron short of a completely full outermost shell. So it's eager to pick up an electron. Again, to pull in an electron, to move an electron from somewhere else, that is a redox reaction. So sodium and chloride together are happily going to participate in a redox reaction, let that electron move from the sodium to the chloride, and you can see it happen when you go from plain old sodium and plain old chloride to positively charged sodium and negatively charged chloride, right? That negative means the electron has moved. You can see the movement of the electron there. This is an in-your-face kind of redox reaction. You can see that change because the electron has moved, and because the electron is moving, there's also a change in charge. The way chemists talk about redox reactions is the sodium is a reducing agent, and the chloride is an oxidizing agent. Typically, in a redox reaction, the oxidizing agent is actually oxygen, right? You'll see that in the next slide. It's pretty obvious. But the thing of it is the oxidizing agent is the thing that pulls an electron to itself, okay? And so here, chlorine, or chloride, is the oxidizing agent, and sodium is the reducing agent. It's the thing that is foisting an electron onto something else. Maybe a high-energy electron getting moved to a low-energy spot, but here that would be sodium. In the process, the reducing agent, sodium, gets oxidized. The oxidizing agent works its magic and oxidizes the reducing agent. And in the process, the oxidizing agent gets reduced because the reducing agent does its thing on it, and you end up with that sort of scenario. So what I've tried to do in talking through this slide just now is actually two things. One, I just wanted you to see an in-your-face kind of redox reaction. An electron got moved from the sodium to the chloride. Bam, there you go. That's a redox reaction. That's what we mean when we're talking about redox reactions. No subtlety to that redox reaction. And the second thing was I wanted to at least say the words out loud to you. They're in the lecture notes as well, and they're in your textbook as well. But I wanted to say the words out loud to you about what a reducing agent is, what an oxidizing agent is. I'll confess, that terminology confuses me, right? When I sit down and I look at the lecture notes, I'm like, okay, remember, that's what a reducing agent is, that's what an oxidizing agent is, right? In a chemistry class, that stuff's going to be really important to you to know, to be really good at whipping those words out and using them in a conversation. In a biology class, it's important to know, but I think you'll get by pretty well just by appreciating, oh, this is a redox reaction. And somewhere in the mix here is an oxidizing agent, and somewhere is a reducing agent. That's probably going to serve you well enough for most biology classes that you'll be taking. All right, so this is an in-your-face redox reaction. The electrons get stripped from this guy and pawned off onto this guy, and you can see that it's happened because there's a full-blown charge there and a full-blown charge there. That's where the electron moved. It moved to where that negative charge is. Here is an example that I flashed at you on Thursday, so the last time we met, that's a more subtle kind of redox reaction. We have, just like in any redox reaction, an oxidizing agent, and I warned you that often, typically actually, the oxidizing agent is actually oxygen. That's how they got the name oxidizing agents, because oxygen is usually involved. And we have a reducing agent. In this circumstance, the reducing agent is methane. It's a more subtle kind of redox reaction than what we had seen on the previous slide. It's more subtle because nothing's got a full-blown charge associated with it at the end of this redox reaction. But if you take a close look at the substrates, or the reactants if you prefer, those words are interchangeable. If you take a close look, what they've done with this dot diagram here is they're showing you there's a covalent bond with that line, but they're also showing you where the electrons fall. Here they're showing you the electrons are kind of like equidistant. They're halfway between the carbon and the hydrogen, right? That's a high energy bond. Okay, if you broke that covalent bond, you'd be releasing a fair bit of energy. These are high energy electrons. And the same is true over here. The electrons involved in the covalent bonds between the two oxygen atoms are also high energy bonds. Putting a different spin on this slide, you guys, based on what we've talked about the last couple of lectures, I hope will realize that there's a high Gibbs free energy associated here. There's a lot of energy stored in these covalent bonds. And therefore, this should be a very favorable, at least from an energetics perspective, chemical reaction. You're going from high energy covalent bonds to low energy covalent bonds. So the delta G should be very favorable. A favorable delta G, a negative delta G, means this is a spontaneous chemical reaction, right? It'll happen of its own accord. You don't have to do anything to help this along. Nature likes this kind of thing. But it's a redox reaction because we're going from high energy electrons like these, and we're moving them to a lower energy state. That's why this counts as a redox reaction. You start off with high energy electrons, and you've moved them to a low energy status. That movement of the electrons is what makes this a redox reaction, even though you don't actually see a change in charge. Methane was the reducing agent. It gets oxidized into carbon dioxide gas, and oxygen gas is the oxidizing agent. It gets reduced to water. Okay? So this is another example of a redox reaction, but it's more subtle. What you need to focus on here is, are the electrons moving? That's always the case if you want to decide if it's a redox reaction or not. And not only are the electrons moving, it's more subtle because here the electrons are moving, but they're moving from a high energy state to a lower energy state. That's what makes it a redox reaction. The redox reaction that's going to be the star of today's lecture and then finished off on Thursday's lecture is the one that I've got up on the screen right now. Okay? This also is a redox reaction. The substrate, or the reactant, that we're really keying in on here is this one right there, right? You guys know from the first part of the class that something that has that kind of formula is a carbohydrate, right? For every carbon, there's one oxygen and two hydrogens. Carbo, C, hydrate, water, right? This is a carbohydrate. And it's a specific carbohydrate that we're going to be talking about for the purposes of today's lecture and Thursday's lecture. It's everybody's favorite sugar, I think, right? That's glucose. I don't know, maybe other people have other favorite sugars, but come on, it's perhaps the best known of all sugars. It's the most famous of all sugars, glucose. All right? So that's the particular 6-carbon sugar that's going to be the star of today's lecture and Thursday's lecture. And the other reactant or substrate is oxygen gas. And I'll tell you, the long and the short of it is this. What we're doing with glycolysis, this whole process here, what we're doing is tantamount to, it's equivalent to taking a sugar cube and holding a match to it and setting it on fire. What's happening in that scenario is you're combining the sugar molecules with oxygen gas in the air to help the combustion take place. And along the way, it burns, and you don't want to hold it because your fingers will get burned. It's hot. You're releasing a lot of energy as you convert the energy in glucose into, and you're combining the glucose with oxygen gas to make carbon dioxide and water, and you're releasing energy in the process. That is the process that we're talking about here. That's another way that process could take place or could play out. Favorable delta G, high energy. Look at the molecules on the left compared to the molecules on the right. The ones on the left are high energy covalent bonds. The ones on the right are low energy covalent bonds, so it's energetically favorable. You're moving electrons, so it's a redox process. They're going from high energy to lower energy. And you could do it by lighting a match and getting the whole thing started that way, or you could do it the way our cells do it and slowly, stepwise, try to extract that energy with enzymes. Question?
So are all redox reactions exothermic?
So the question is are redox reactions exothermic, releasing heat? I don't think that they have to be. I don't know that that's part of the definition. Certainly this one is, right? And certainly the one on the previous slide where we're igniting methane is. And actually the one on the slide before that where we're lighting hydrogen and oxygen gas together are. So, we've got 3 for 3 here, but I don't know that that's a central rule. The rule is you're moving electrons from high energy states to low energy states, or just moving them, period. Okay, so you got a choice. You could do this in one big poof, or you can do it in a controlled fashion and extract that energy to do something useful with. In the lecture notes, I give you another option here. We've got a lot of high energy bonds in the petrochemicals inside of our gas tanks for our cars, right? You can get your car to move by exploding that gas tank, right? It'll move. Probably won't move anymore after you've done that explosion of the gas in your gas tank. Or you can do it in a controlled process to get that motion to take place and do it in a way that's perhaps more useful to you. Obviously, one of those two is a better option than the other, right? And it shouldn't surprise us to see that living things over the course of billions of years of evolution have opted for the more controlled process as opposed to that quick, immediate release. But again, this is what it is that we're going to be talking about. And we're going to talk about today the first half of this process, going from glucose to 2 3-carbon sugars that then get broken down into 6 1-carbon molecules, carbon dioxide gas. That process, getting from the 3-carbon sugars to the 1-carbon molecule, carbon dioxide gas, that's what Thursday's lecture is going to be about. Today we're going to focus on the first half, getting from 6 carbons down to 3 carbon molecules. Interesting typo there. There's a missing N from the end of that word. I wonder if we'll see that again. I bet we do. But I want to draw your attention before we dive into those chemical reactions and talk about the enzymes that are involved. I want to draw your attention to the fact that what I'm going to be talking with you about today is actually an anaerobic process. An is the Greek prefix for without, right? Aerobic is a reference to oxygen. So the process that I'm going to talk with you about today is anaerobic. It can take place in the absence of oxygen, okay? And that's in contrast to an aerobic process, one where oxygen is present and plays an active role in the process, okay? On Thursday, I'll talk with you about aerobic respiration, how we can link the burning of glucose directly to oxygen to extract every last little bit of energy out of it that we can. Today, though, we're going to talk about an anaerobic process. There's no oxygen required or involved. Here's the thing. If you don't use oxygen, for goodness sakes, oxygen is an exceptionally good reducing agent, right? Or an oxidizing agent. Oxygen is like the be-all and end-all of redox reactions. It sort of sets the bar for everything else, because you go from this high-energy molecule, oxygen gas, to ultimately having low-energy molecules like water. Water is kind of like the definition of the low-energy molecules. If you're not making water out of oxygen gas, you're probably leaving some energy on the table in the form of some covalent bonds that you haven't extracted the energy from. So you really want to get as much bang for your buck out of the covalent bonds of a glucose molecule. You want to do aerobic respiration. But there are organisms, and there are circumstances, even for organisms like ourselves, where aerobic respiration isn't possible, right? Anaerobic exercise, that's what that means, is that you're doing stuff faster than you can bring oxygen to your muscles to get the whole, process of glycolysis finished off. So there are times that even you and I have to deal with just the anaerobic respiration. And there are organisms, botulism comes quickly to mind, that are adapted to living in the absence of oxygen. The best that those organisms can do is anaerobic respiration. And the long and the short of it really is this: anaerobic respiration doesn't let you extract nearly as much energy as aerobic respiration does. The end products of anaerobic respiration still have a lot of high energy covalent bonds. You're leaving energy on the table if you're playing a game here. And that doesn't happen with anaerobic respiration, with aerobic respiration. With aerobic respiration, at the end of the process, all that's left is carbon dioxide gas and water, two end products that have very little energy associated with their covalent bonds. So I've said, anaerobic respiration doesn't get as much energy out as aerobic respiration does. Here's the actual numbers to it. The anaerobic respiration we're going to be talking about today gives cells a net gain of 2 ATP molecules. For each glucose molecule that gets broken up, you get 2 ATP molecules. With aerobic respiration, the net gain that you get is not just those 2, you add 32 more on top of that. So you get a total of about 34 ATPs from aerobic respiration by milking every last bit of energy you can out of that energy-rich glucose molecule, as opposed to the 2 that you get if the end of the road for you is pyruvate. Aerobic respiration gets you a lot more energy than anaerobic respiration, 2 versus 34 ATPs. The 2 ATPs is hideously inefficient. I've been joking for the last couple lectures about this idea of the currency exchange, taking a $100 bill to the bank, and how much will you give me for this $100 bill in singles? All right. For anaerobic respiration, the payoff that you get for your $100 bill, 686 kcals per mole, you get 14.3 kcals per mole. In terms of dollars and cents, you go to Right Path Credit Union, you give them the $100 bill, I want as much change for this as you'll give me. They give you $2.10 back. 2.1% efficient in terms of the conversion, the extraction of the energy from glucose into useful form of energy in ATP. So anaerobic respiration, it's hideously inefficient. We were looking down our noses at how inefficient aerobic respiration was, where you get 34 ATPs made. That's only, what, about 35, 36% efficient. We thought that was bad. How much worse is 2.1% efficiency? Yet that's what anaerobic respiration does for you. And that's what it is we're going to talk about today. You know, I don't think anybody's arguing anaerobic respiration's a good idea, but if you don't have oxygen and that's all you can do, 2% efficiency is better than not having any energy at all. That's the price that things pay when they have to go or they decide to go anaerobic. All right, enough about anaerobic versus aerobic. Here's what's going to happen. Let's talk about glycolysis. Here is glycolysis from a very high level perspective, broad brushstrokes. What we're talking about with glycolysis is pretty simply this. We start with glucose. That's our substrate or our reactant. And at the end of glycolysis, we make 2 pyruvate molecules, and we create a net gain of 2 ATPs plus this other thing called NADH. We make 2 NADHs. That's it in a nutshell. That's what the rest of today's lecture is about, is that. Okay? How could I possibly stretch another 30, 40 minutes of talk out of something like this? You guys are savvy. You know how this is going to play out now. Guess what? There's a lot of stuff going on in this arrow. Okay, so that's going to account for a fair bit of our time for the rest of the day today. I'm going to break this arrow down for you and talk about how it is that things are playing out in each of the steps along the way in this arrow. That arrow, in a word, gets called glycolysis. Right? That's the process of converting glucose to pyruvate. Again, 6-carbon sugar, 3 carbon sugars. One of the things that's kind of helpful to students as you're trying to wrap your head around this business is keep a good accounting. Be an accountant for a little while with regard to the carbon atoms. Here you had 6. Here you've got 2 times 3. At the end, when we get to talking about aerobic respiration, what's going to happen is you're going to start with 6, you're going to get 2 3-carbon things, and at the end you're going to end up with 6 1-carbon things. So keep nose counting the carbons. That'll help along the way, and that becomes particularly helpful when we start talking about photosynthesis. It's the flip side of glycolysis, how you make sugars as opposed to breaking them down. And let me also, just for a minute, say a little bit more to you about that NADH today. We'll talk about it a fair bit more on Thursday. For today, NADH is a bit player. It's just making a cameo appearance and goes off. For things that are stuck with doing anaerobic respiration, NADH is actually a problem. You have to get rid of the high energy electron associated with the proton that is the H in NADH, right? That actually ends up being a problem. You got to figure out some way to dish that thing off and get rid of it in anaerobic respiration. But with aerobic respiration, that NADH is actually where the money is, because you can use that hydrogen on the NADH, the H of the NADH, to drive a process whereby you can get some serious ATP. That's the main player, it turns out, in aerobic respiration. We'll talk about that more on Thursday. For today, NADH gets made. And that could be the end of the story if you're talking about an anaerobic process, but there's more to the story if you're talking about an aerobic process. For today, let's just keep our eye on 3 balls. We're talking here about a sugar being made into 2 smaller sugars and the release of some energy. There's no point for an organism doing glycolysis if not for the fact that it needs ATP. The whole point of glycolysis is you're trying to make some ATP molecules so that you can hook those ATP molecules up to other chemical reactions to make them more energetically favorable. That's the whole point. So those 3 things then are pretty obviously going to be the most important things to keep track of here. Perhaps the single most important thing is how are we actually going to make ATP along the way. Again, the process is called glycolysis. There are 10 enzymes involved in glycolysis. Here's a closer look at what's happening in that arrow. And before we're done, we're going to take a closer look still. So let me just, pace yourself in terms of your enthusiasm for what's going on here today. We're going to get into deeper and deeper detail as we go. So, glycolysis. Start with glucose, end with pyruvate. You make some ATPs. Big picture, how this plays out is this. Again, you start with glucose. You actually have to prime the pump with investing some energy at the beginning of the process of glycolysis. When I used the word you in the previous sentence, I literally meant you, right? But I also want to make sure you appreciate it's everything. All living things do this process exactly the same way. We use the same enzymes, we use the same strategies. Bacteria do it, plants do it, your dog does it, you do it. We all do this exactly the same way. And that general strategy that we all use is we start by investing some energy. We juice up the glucose molecule, which again, for goodness sakes, it's pretty energy rich to begin with. We make it even more energy rich by attaching some phosphate groups to it. And then, after we've really got the glucose all quivery, full of energy, then we release it. And we release it in a stepwise process that gives us a total of 4 ATPs being made, as well as those 2 NADHs and pyruvate and water. The net for this whole process is you invest 2 ATPs to get 4 ATPs out, so you have a total gain of 2 ATPs going from glucose to pyruvate, and you also, in some sense, have a total gain of 2 NADHs made from NAD+. And of course, the glucose ends up being 2 pyruvates and water. So this slide shows you a little more detail of what's going on. The new thing in this slide relative to the previous slide is you see you start with an investment of energy. You prime the pump before you actually start to extract that energy. That's the new piece of information you got from this slide relative to what we've talked about before. Now, remember, when a chemist, and I suppose when a biochemist puts an arrow somewhere, that means there's a chemical reaction that's taking place. All those chemical reactions get names. In a living system, for metabolism, all of those chemical reactions are catalyzed by a biological catalyst. All of those are catalyzed by enzymes. So a lot of the rest of today's lecture is going to be simply my showing you the chemical reaction that gets catalyzed at each step along the way, telling you the name of the enzyme, and reminding you that you start by investing energy and then you start extracting energy. All right? That's my job for the next 20 minutes or so, is to do that. We're going to look at each of those arrows. We're going to break them down. I'll tell you the name of the enzyme. I'll tell you that, hey, look at this. We invested some energy. Hey, look at this. We're extracting some energy. That's what you're in for better or worse. So let's do it. Let's get it over with. Let's rip that Band-Aid off and see what's actually going on here. I'll ease you into it a little bit. Your textbook uses a particular color coding convention here. Whenever you see this, and I'm so thankful that they tell me the name of the color to call this. Whenever you see this teal color on a figure that I'm showing you on these slides or in your textbook, that means we're talking about glycolysis. It's color consistent. Whenever you see, I would have called this pink, but it turns out it's actually salmon, right? Whenever you see the salmon color, that means what's happening there is pyruvate oxidation in the citric acid cycle. That's what we're going to talk about on Thursday. And whenever you see this, again, purple, apparently it's actually violet. Whenever you see this color, that means we're talking about something that is unequivocally oxygen dependent. This is aerobic respiration, oxidative phosphorylation. That also is something we'll talk about on Thursday when we talk about the final process of glycolysis. Most of today is going to be this teal-colored stuff. Where glycolysis takes place inside of a eukaryotic cell is pretty compartmentalized, right? We've got these compartments, and we use them to help this process of glycolysis. The business we're going to talk about today, going from glucose to pyruvate, happens exclusively in the cytoplasm, period. It's in the cytoplasm. And that also then would apply to prokaryotes and archaebacteria. Glycolysis, going from glucose to pyruvate, just happens in the cytoplasm. For eukaryotes that have these internal compartments, the mitochondria, the rest of it takes place there. So pyruvate oxidation, the citric acid cycle, all of that salmon-colored stuff, takes place exclusively inside the mitochondria. And also, exclusively in the mitochondria is this business of oxidative phosphorylation. I'll tell you right now, that's where you get the most bang for your buck. That's where most of the energy gets extracted, is the oxidative phosphorylation part. You can't do that unless you got a mitochondria to help you pull that process off. It gives us a huge advantage over things that don't have mitochondria to work with. So that's the big picture. But again, the chapter of this story we're focused on today is this arrow here, the business of glycolysis. So we're going from glucose to pyruvate. You start with an energy investment part before you can do the harvesting. Here it is one last time. gentle, easy, high-level view of what's happening here. It's a 10-step process. There's the first step in the process. All right, so back in the old days, when I was a freshman in a biology class, the mean old faculty member made me memorize every last bit of this, right? It was particularly hard after having to walk uphill 2 miles to get to school, right? And knowing that I still had another 2-mile walk back home uphill as well, right? In the snow, it was always snowing and it was miserable. I'm looking for sympathy here. It was hard in the old days. We had to I was forced to memorize all of this stuff. The structures of the molecules, the names of the enzymes, the whole pathway. Took hours. It was painful. There's a part of me that wants to haze you that in the same way that I got hazed myself, right? But I'm a nice guy. I've suffered. I don't feel you guys need to suffer. I'm not going to demand that you guys memorize this whole process the way that I had to. There's a good chance that in the next classes that you take in a biology curriculum, they're going to scold you for not having memorized this stuff. How could you have not done that? And they'll get back to me and say, why didn't you make them memorize it? And I'll say, because I wanted you to be the mean person, and I want to be the cool, nice guy that gets the good student evaluations at the end of the year. Right? That's what this is all about for me. But I'm warning you, there are going to be people that you encounter during the rest of your academic careers that will insist you memorize this stuff. I'm not that guy. Somebody else will. If you want to memorize it, go for it. You'll be in good shape down the line. Not a problem. But there are things that I do want you to notice as we go through this process. And I'm going to call them out to you. I'm going to help you feel good about those things. Don't let me down on those things, right? Here's the important stuff. I'd like for you to be able to recognize glucose. If somebody flashed a structure of glucose up at you, like what you see in the left side of the screen there, it's like, oh, that's glucose. You're going to see glucose structures a lot. I'll tell you, I'm not that worried that you know if the OH is on the top or on the bottom at each of those points along the circle. But boy, come on, is it asking that much for you to recognize that that's a 6-carbon molecule, right? If you see somebody talking about a 6-carbon molecule in a biology class, they're almost certainly talking about glucose. And if they're not talking about glucose, they'll probably tell you this is maltose or fructose or something else, right? So check it out. That's glucose. There's a carbon here. There's carbon here, 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 here. And this one's obvious, here. 1, 2, 3, 4, 5, 6. So we start with a 6-carbon molecule. It's glucose. That's the way people usually depict it. That's the way they'll show it as a typical picture. So that's one thing. Know what glucose looks like. At the end of today, I'm going to say, look at that. That's pyruvate. Kind of know what pyruvate looks like. Know what it starts like, know what it ends like, and we're cool. That's one thing. Don't worry too much about the structures in between glucose and pyruvate. With this exception, try to make sure you can tell when you see a structure along the way here, which structures have the most energy associated with them. All right? Do that business of counting high energy bonds. For your bookkeeping, I've got a new wrinkle to throw at you. Count the number of high energy bonds. And by that I mean carbon to carbon or carbon to hydrogen. Those count equally and count as one. But here's the new wrinkle. If you see a phosphate group attached to something, assume that means it's getting more energy from the phosphate group being there. Okay? In the scheme of things, it's probably worth another covalent bond or two. Don't worry too much about the particular. But when you're looking at these structures, lots of carbon to carbon and carbon to hydrogen bonds, high energy. Phosphate group, higher energy still. Fewer carbon to carbon and carbon to hydrogen bonds, and no phosphate groups, less energy. And you're going to see the whole gamut in between. see if you can't be comfortable with rank ordering them. This is the most energy. Well, this one's got a lot of energy. This one's got even more energy. This one's got a little bit less energy, about the same, less, right? That's the kind of thing I'm hoping you'll take away as part of the big picture here. So recognize glucose and pyruvate. That'd be nice. Be able to identify energy-rich molecules relative to energy-poor molecules and see how they fall out relative to each other. Develop an eye for figuring that out. It's not that hard, but it comes in really handy, not just in this class, but others. And then here's the other thing. Do not get freaked out or intimidated in any way by the names of the enzymes that catalyze these reactions. There is a real tendency for faculty members to try to intimidate you with these names. There's a real tendency for you guys to be intimidated by the names. Don't be intimidated by the names. Part of the name is always either the substrate or the product of the reaction, but the other part of the name tells you what kind of enzyme we're talking about. And there are essentially only 3 kinds of enzymes that we're going to talk about, and I'd like for you to know what each of those 3 kinds of enzymes does. Okay? One of those kinds of enzymes are kinases. And I'll show you. There's a kinase right there at the start. A kinase is an enzyme that transfers a phosphate group from one thing to another thing. Period. All kinases involve the transfer of a phosphate group. You're going to meet a couple of kinases during the course of the next 10, 15 minutes or so. Don't let the word kinase freak you out. It just means that this is an enzyme that's transferring a phosphate group from one thing to another thing. That's it. You're also going to meet enzymes, another class of enzymes, that change the shape of a molecule, okay, from one thing to another thing. In a word, they're going to change something from one isomer to another version of itself, another isomer. You know what we call those enzymes? Isomerases. Okay? So if an enzyme's got isomerase in its name, all that means is it's involved in changing the structure of the molecule from one isomer to another. Somewhere along the way, one of these enzymes was named by a guy who didn't appreciate that convention, and he used the word mutase instead of isomerase. Mutase and isomerase are kind of interchangeable. So we've got 2 different classes of enzymes now. We've got the kinases, we've got the isomerases, parenthetically mutases, and then the third kind of enzyme that you're going to meet during this glycolytic process are the dehydrogenases. All right? That's it. There's only 3 kinds of enzymes that are involved in this business: kinase, isomerase, and dehydrogenase. You'll never see a dehydrogenase do anything unless NAD or NADH are involved. Okay? That's what they're all about. Dehydrogenases are all about moving that hydrogen with an energy-rich electron onto NAD or moving that hydrogen with its energy-rich electron off of NADA. And dehydrogenases are always just moving that hydrogen onto or off of NAD. Don't worry about the names of the chemicals as you go from the beginning to the end. I'd like for you to know the first, glucose, and I'd like for you to know the last, pyruvate. Don't be too worried about the names of the molecules in between. That's part of the names of these enzymes. Don't worry too much about that, but be comfortable with the idea of what a kinase is, a mutase or an isomerase is, and a dehydrogenase. So there's my best shot at making you at ease with what you're going to see, because without that, I can see how this could be awfully horrifying. It was to me. But here's what's going on. You start out with glucose. You guys all recognize glucose. I asked you to be able to recognize glucose. That's glucose. You start out with glucose. And we've just been talking about how you start the business of glycolysis by investing some energy. into the glucose molecule itself. If you want to invest some energy, we've been talking for weeks now about ATPs being the dollar bill of the energy currency of a cell. What that means now in this context is we need to get some of the energy out of an ATP molecule and tack it onto, transfer it over to a glucose molecule. The tangible representation of the transfer of that energy has got to be putting a phosphate group onto glucose. So check it out. The first step in glycolysis, it's a 10-step process, but the first step involves transferring a phosphate group from ATP to glucose and releasing ADP, a less energy-rich thing, as a byproduct. We're investing some energy. The name of the enzyme that does that particular catalysis, that catalyzes that particular reaction, I sure hope it doesn't surprise you at this point to hear that it's a kinase. And it's everybody's favorite kinase, hexokinase. You know where it gets the name hexo from? I told you enzymes get their names. There's two parts to the name. There's what it does. That's the second part, the kinase. And the first part is the name of one of its substrates or its product. Hexo here is a tip of the hat to glucose. Hex is 6, right? It's a 6-carbon sugar. You want to be really, I don't know. You guys decide how you want to be. If you want to say, if somebody's talking with you about glucose and you stopped them and you said, I prefer to refer to glucose as hexose, right? I'm a hexose kind of person, not a glucose kind of person. They're going to think immediately that you're a jerk, but that's also correct. Some strange people don't like to use the word glucose. It rubs them the wrong way for some reason, and instead they'll say hexose. All right? It's an equivalent term. There aren't many people like that left in the world because they've been identified as jerks and other bad things happen to them as a result. But that's what this enzyme's name is all about. It is an enzyme that attaches a phosphate group onto glucose. That's hexokinase. Check it out. You started off with ATP, you end up with ADP, and check it out. The glucose isn't glucose after hexokinase does its thing. Now it's got a new name. It's called glucose-6-phosphate. There's the structure of glucose-6-phosphate. The name isn't that mystifying, right? Nobody should be too worried about glucose. That shouldn't scare anybody. 6-phosphate, you know what? There's a phosphate group on this glucose molecule that wasn't on this glucose molecule. Where's that phosphate group? Oh, wait, it's on the 6th carbon. Carbon 1, 2, 3, 4, 5, 6. Where's the phosphate group? It's on 6, hence the name glucose-6-phosphate. Nothing too scary. Maybe you're thinking, I want to memorize this. Right? This isn't hard. Wait. All right. So there you go, glucose-6-phosphate. 10-step process. We're done with step number 1. Let's go on to step number 2. Now what's happened? Well, there's an enzyme called phosphoglucoisomerase. Let's break the name down. Phosphogluco. I know what they're talking about. This is glucose-6-phosphate. Phosphoglucoisomerase. This is the name of one. It's either the substrate or the product. It's apparently the substrate. This enzyme is going to do something with this molecule. What's it going to do with that molecule? The second part of the name is isomerase. Kinases move phosphate groups around. Isomerases make isomers. They change the structure of the molecule. What do you suppose phosphoglucoisomerase is going to do? It's going to rearrange some of these covalent bonds. There's no difference in the amount of energy associated with this structure versus this structure. But now instead of glucose-6-phosphate, because of the arrangement of those covalent bonds, it's a new compound. It's called fructose-6-phosphate. This is fructose. It has a phosphate group attached to it, fructose-6-phosphate. Let's get some rubber hitting the road here. If on an exam I said to you, which of these 3 molecules has the least energy associated with it? The winner is glucose. You know how you would recognize that? It's the only thing here that doesn't have the phosphate group on it. Everything else is the same. The number of covalent high-energy bonds, carbon to carbon, carbon to hydrogen, same here, same here, same here. This one doesn't have a phosphate. These two do. Which of these two has more energy associated with it? It's a push. I don't know. They're the same. My accounting system doesn't help, right? They have the same number of carbon to carbon and carbon to hydrogen bonds, the same number of phosphate groups. It's the same. I can tell you, I don't need you guys to necessarily appreciate this, but I can tell you, if I had to put a number on it, I'd say that this one has less energy than this one, just because to go from here to here, it has to be an energetically favorable reaction. So there's probably a little bit less energy, but it's probably a real little bit less energy. But because it's a spontaneous reaction, it's got a favorable delta G, I'm feeling pretty good about saying this probably has a little less energy than that, but that's like sophomore level biology, not introductory biology like what we're talking about here. So moving on. Look at that. We cruised through number 2, right? 1 took us a while, 2 wasn't that bad. What's going to happen for number 3? This is our substrate. What's our product? Well, the enzyme involved is a kinase. That should telegraph to you, it should shout out to you, a phosphate group's going to get moved around. Where's the phosphate group going to move from and to? Right? Well, I've been saying at the beginning we're investing energy. I've said we have to burn 2 ATPs to get the pump primed. This is consistent with that story. We're moving a phosphate from ATP onto the sugar that is the substrate for this reaction. So phosphofructokinase is moving a phosphate group in this energy investment phase from ATP onto fructose 6-phosphate. And you end up with fructose, because that hasn't changed, but now it's got 2 phosphates. Bis is another way of saying 2 phosphates. So you get fructose 1,6-bisphosphate.
So why doesn't it use the ADP still has 2 phosphates? Why doesn't it just use another phosphate?
So the question is, why do you keep going to the well with ATP instead of getting that extra energy you could out of the ADP? Because there is energy in the ADP. I don't know the philosophical answer to the why, but I can tell you the practical answer, and that is that every opportunity cells have to convert ATP to ADP as opposed to ADP to AMP, they do. Cells only get that ADP to AMP into gear when they're really hurting for energy. That's like their last resort to get energy there. So for whatever reason, philosophically, they don't want to do that. It's, I don't know, misanthropomorphically, it's like, I'd rather break my $100 bills into singles, even though I could break 20s into singles. I really want to do the hundreds. I don't know, there's something like that going on. All right? Not a particularly satisfying answer, but that's just the way it works out. Okay, God likes it that way. How about that? That'll work. Okay? So, check out what's happened here. We got an enzyme, out of context, without the gentle introduction that you've had, phosphofructokinase might scare you a little bit. But it shouldn't be that scary. Part of the name is what the enzyme does, kinase. It's moving a phosphate group from one thing to another thing. And the other part of the name is usually the first part of the name is either the substrate or the product. Here it's the substrate. Fructose 6-phosphate is the substrate. Phospho, tip of the hat to the phosphate. Fructo, tip of the hat to fructose. Phosphofructokinase. Again, this is an energy investment process. We're still moving energy into glucose. We're expending ATPs. We're not making ATPs. We're losing ATPs in the hope that by the end of this process, we'll get a net gain of ATPs. You can put a lot of faith in that hope because it's going to happen. What does the fructose 1,6-bisphosphate look like? It looks an awful lot like fructose with 2 phosphate groups on it, as opposed to fructose with 1 phosphate group on it. If I were to ask you which of these four molecules has the most energy associated with it, this one, this one, this one, or this one, I'm hoping you guys go with this one, right? My accounting method works pretty well. A lot of carbon to carbon, carbon to hydrogen bonds, but there are now two phosphate groups. Two phosphate groups beats one phosphate group. This is the winner. Simple. You guys want to memorize it? It's not that hard. You can do it. Go for it. Have fun. Impress your friends. Because I got to tell you, if you thought the reticuloendothelial system helped you in the romance department, any one of these enzyme names is gold, right? Have fun with that. Okay, so we've been doing energy investment. When are we actually going to start to get something out of this? Not quite yet. We've got to bust this molecule now up into something that's a little bit more manageable. We're going to break the 6-carbon fructose molecule into 2 3-carbon molecules. If you cut it right down the middle, you get dihydroxyacetone phosphate. There's a name that only a mother could love. And you get glyceraldehyde 3-phosphate. All the action turns out to be with glyceraldehyde 3-phosphate. This is what ends up getting channeled through the rest of the process. So you got to convert the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. Check it out. An isomerase does that. You guys know what isomerases do. This one is such an important isomerase, so centrally important to life. It's just called isomerase. It is the paradigm, the epitome of isomerases. It goes by just one name. It's the share of glycolysis isomerase, period. Okay? Don't worry about aldolase. That doesn't conform with any of my rules. Okay, so you bust up the fructose 1,6-bisphosphate into 2 3-carbon things, each with a phosphate group, each with an equivalent amount of energy, each of which can be interconverted with the other through the catalytic activity of an enzyme called isomerase. Ultimately, all of the dihydroxyacetone phosphate gets made into glyceraldehyde phosphate because there's a pipeline here. These molecules are also getting shunted into the rest of glycolysis. So you lose these guys, and they get replaced to some extent by these guys getting converted over to there. And there's fewer of those to go back this way, so it's a net flow in this direction. We've gone through half of glycolysis. There are 5 enzymes involved, 10 enzymes involved. We've talked about 5 of them. Let's get out of energy investment mode and start getting the payoff. Energy payoff. First step in the energy payoff mode is a dehydrogenase gets involved. I told you at the beginning, dehydrogenases always move a hydrogen either onto or off of NAD. Okay? This particular dehydrogenase is taking a hydrogen off of glyceraldehyde 3-phosphate and onto NAD. And it's not so much taking a hydrogen so much as a high energy electron. The hydrogen comes from somewhere else. And it also manages to, in the part of the process, bring in phosphate groups. So triose is a tip of the hat to the 3-carbon sugar, glyceraldehyde 3-phosphate. So triose phosphate. Go back to that last picture. 3-carbon sugar with a phosphate, triose phosphate if you like. And a dehydrogenase makes an NADH at the same time that it takes inorganic phosphate, not from ATP, and it tacks it onto that 3-carbon molecule to make 1,3-bisphosphoglycerate. Pretty energy-rich 3-carbon sugar. Okay, so in some sense, this is an energy payoff, only in the sense that that NADH can be used to drive the synthesis of ATP through oxidative phosphorylation. In an anaerobic environment, you can't do oxidative phosphorylation. Just write that off. It's gone. It's over. You haven't really gotten an energy payoff at that stage. But in an anaerobic environment, if you got working mitochondria, you can get some pretty good ATPs off of the NADH. That's why it shows up in the payoff phase. But now we're ready to start actually making some ATPs. There's two ways that we can talk about making ATPs. There's two different strategies for making ATPs. There's the oxidative phosphorylation that we'll talk about on Thursday as it plays out in the mitochondria. And then the alternative is what's called substrate level phosphorylation. For substrate level phosphorylation, you've got an enzyme that's literally moving one of these phosphate groups onto ADP to make ATP directly. Okay? The next step is an example of substrate level phosphorylation. If you didn't listen to what I just said for the last 30 seconds, here's a picture that summarizes what I just said. For substrate level phosphorylation, you get an enzyme to cuddle up with a substrate that's got a phosphate group on it, bring that into close proximity with ADP, smush the two of them together, and release a product that doesn't have a phosphate group and ATP. Okay? That's substrate level phosphorylation. The next enzyme in glycolysis, enzyme number 7, is phosphoglycerokinase. Kinase because it's moving a phosphate group from one thing to another, from the substrate onto ADP. Phosphoglycero because it's starting with 1,3-bisphosphoglycerate. All right, two-part name. And another substrate here, don't forget, is ADP because one of the products will be ATP. Did you notice that there's 2 ADPs and 2 ATPs, and that there's 2 NADs and 2 NADHs? Let me just make sure you understand why that 2 is there. It's because there's 2 ATPs involved, right? There are 2 ATPs involved because, remember, we started with a 6-carbon molecule, and we're doing this to 2 3-carbon molecules. You broke that 6-carbon thing up into 2 3-carbon things. Each of the 3-carbon things gets the same treatment. 2 ATPs are getting made here. We got to do a little bit more moving stuff around. I told you this guy, when they named this enzyme, they forgot that most people like to talk about isomerases for doing this sort of thing. Mutase, isomerase, potato, it's kind of the same thing. But what's going on here is 3 phosphoglycerates getting changed a little bit. So phosphoglyceromutase converts 3-phosphoglycerate into 2-phosphoglycerate. All that's happened is you moved the phosphate group from the end of the molecule to the middle. All right? Literally, that's all that's happened. That's enzyme number 8. We're in the home stretch. Hang with me for 2 more because the 9th one is an enolase. Don't worry about that. We're kicking off a water molecule. There's not a lot of energy associated with that, right? It's almost a bookkeeping thing, but we make phosphoenolpyruvate. And ultimately, pyruvate kinase comes along to finish this process off. Another instance of substrate-level phosphorylation because pyruvate kinase cuddles with phosphoenolpyruvate, brings ADP to the party, smushes the 2 of them together, and you make ATP and pyruvate. Glycolysis is this business of going from glucose to 2 pyruvates. We've accomplished that. Notice that we had to invest 2 ATPs to get the process started, but that along the way, we've also, through substrate-level phosphorylation, made 4 ATPs, net gain of 2 ATPs. 2 invested, 4 returned, net gain of 2 ATPs. That's glycolysis. 10-step process catalyzed by 3 different kinds of enzymes, kinases, dehydrogenases, and isomerases. And here's a closer picture of all of those processes. Right. I don't need to say that anymore. Remember this, we dove in deep, now we're pulling back out. This is what we started with. You got the investment phase, you get the payoff phase, you get a net gain of 2 ATPs and 2 NADHs. Don't forget, you started with glucose, you ended with pyruvate, and you made some water along the way. So that's glycolysis. To get the rest of the energy out of a glucose molecule, you need to invoke something else. It's called the Krebs cycle, named for the guy who discovered it and really characterized it. He, in modesty, wanted people to call it the citric acid cycle. You'll see why citric acid might make sense when we talk about that more on Thursday. But the first step in that process is simply this. Remember, glycolysis is taking place in the cytoplasm. The Krebs cycle, the citric acid cycle, that takes place in the mitochondria. So you got to get the energy-rich sugar thing from the cytoplasm into the mitochondria. That's pretty much where we're going to start on Thursday. We're going to move the pyruvate from the cytoplasm into the mitochondria. Along the way, that releases our first instance of a one-carbon molecule in this process. It converts the pyruvate into acetyl-CoA and lets us carbon dioxide go free. That's the end of the story for today. Hey, for the student evaluations at the end of the year, remember that I let you go 3 minutes early this time, right? Don't forget about that. Don't be telling me I always keep you late. Today we're 3 minutes early. See you guys Thursday.